In this video, we're going to be looking at the effect of endogenous pacemakers and exogenous Zeitgebers on the sleep-wake cycle. So endogenous pacemakers, these are internal body clocks that regulate many of our biological rhythms, such as the influence of the suprachiasmatic nucleus on the sleep-wake cycle, which we'll look at in more detail. Exogenous Zeitgebers, these are external factors that affect or entrain our biological rhythms, such as the influence of light on the sleep-wake cycle. So the key endogenous pacemaker we're going to be looking at is the suprachiasmatic nucleus, or the SCN. The SCN is a tiny bundle of nerve fibres located in the hypothalamus in each hemisphere. It is one of the primary endogenous pacemakers in mammals, including humans, and it's really influential in maintaining circadian rhythms such as the sleep-wake cycle. So we're going to be having a look at this process on the next slide. I think I've got diagrams and then I've got a flow chart to kind of illustrate this. Okay, so I'm going to take a look at this diagram first of all. So we can see here that there are nerve fibres which are connecting each hemisphere here to our retina. So our retina receives light information. And nerve fibres connected to the retina in each eye cross over on their way to the occipital lobes and the primary visual cortex. They cross over at a point here called the optic chiasm. The SCN, the suprachiasmatic nucleus, is located just above the optic chiasm. Hence why it's called the supra, which is meaning above, chiasmatic, the optic chiasm. So the suprachiasmatic nucleus is located above the optic chiasm, the point where light information crosses over. And therefore, the SCN receives information about light directly from the optic chiasm. And this happens even when our eyes are closed, which is obviously necessary so that while we're asleep and our eyes are closed, our SCN is still able to receive light information. So it doesn't rely on our eyes to send that light information. Okay, the other key thing that we need to know here is that the suprachiasmatic nucleus is connected to the pineal gland via neural pathways. So let's take a look then at how the suprachiasmatic nucleus helps to regulate and influence our sleep-wake cycle. So at nighttime, there are low levels of light. This information is received by the retina at the back of each eye. This is where our cones and rods are located. There are then neural pathways that send this light information to the suprachiasmatic nucleus. So while that light information is crossing over via the optic chiasm, the suprachiasmatic nucleus is receiving this information about low levels of light. So there are neural pathways that connect the suprachiasmatic nucleus to the pineal gland. If we go back to this diagram, we can see that the suprachiasmatic nucleus located here is connected to the pineal gland here. So there are neural pathways that connect the two, which means that our pineal gland now receives information about levels of light. When there are low levels of light, the pineal gland is instructed then to release melatonin. Melatonin, you will know if you've ever taken it to help you sleep, causes drowsiness and helps to induce sleep. So this is how the SCN helps to regulate the sleep-wake cycle. When there's low levels of light, that information is received by each retina. The suprachiasmatic nucleus is then going to receive that information via these neural pathways. There are further pathways that connect this SCN to the pineal gland. When there are low levels of light, the pineal gland will release melatonin, which induces drowsiness and sleep. And this is why at nighttime we feel sleepy, and then during the daytime we feel more awake. When there are high levels of light during the day, this information is received by each retina. There are neural pathways which take this light information to the suprachiasmatic nucleus, which will then forward this to the pineal gland. And when there's high levels of light, then we're going to suppress levels of melatonin, and this is going to reduce drowsiness and promote wakefulness instead. So this is how the SCN regulates our sleep-wake cycle. Okay, so let's have a look at evaluating endogenous pacemakers and in particular the SCN. So we're going to be looking at supportive evidence from animal studies. So in this research by DeCourcy in 2000, they destroyed the SCN connections in the brains of 30 chipmunks. They then returned these chipmunks to their natural habitat and observed them for 80 days. They found that the sleep-wake cycle of the chipmunks disappeared. So they were still getting the same amount of sleep, but they weren't showing this cyclical pattern that we should expect to see in a normal sleep-wake cycle of mammals. By the end of the study, a significant portion of these chipmunks had been killed by predators, presumably because they were awake and vulnerable to attack when they should have been asleep. So that shows us that the SCN is absolutely critical in establishing and maintaining this sleep-wake cycle. But of course, if we're going to be relying on chipmunk studies to provide us evidence of the importance of endogenous pacemakers and the SCN, then we need to consider two things. One, we need to consider whether it's ethical, and two, we need to consider whether we can even generalize from these animals anyway. Let's start with the ethics. Some might argue that research into the SCN by causing damage and potentially making chipmunks vulnerable is justified. Yes, they were exposed to extreme harm, but they have provided us with really important findings about the importance of the SCN. Now, it wouldn't be possible for us to intentionally damage the SCN with human patients, and it's incredibly hard to find people that have already suffered trauma specifically to the SCN. So we need animal studies to provide these sorts of insights. But does that justify such adverse procedures? Does that justify intentionally harming, knowing that they may be killed by predators? That is a cost-benefit analysis that we need to really perform. The other question that we need to ask is, can we generalize the findings from animals to humans? Yes, we can. Animal studies are typically justified here because there are very similar mechanisms at work across species here. The existence of the SCN and the pineal gland in chipmunks means that generalizations can be made to the human brain, as the mammalian brain has really similar structures. But is it okay to expose these hamsters to risk when returned to the natural habitat, knowing that a lot die as a result? So does what we learn from these studies really justify the ethical procedures? That is a question for you to answer. Okay, so that is endogenous pacemakers. Let's take a look now at exogenous Zeitgebers. The word Zeitgeber is a German word meaning time giver. Exogenous Zeitgebers, these are external factors in our environment that reset our body clock through a process known as entrainment. Basically, they keep our body clock in line. So while we've got our endogenous pacemakers that are keeping things ticking along internally, exogenous Zeitgebers help to sort of reset them or keep them in line when our environment changes. And we have seen from Seif's cave study and other bunker studies that without these external cues, without these exogenous Zeitgebers, the free-running sleep-wake cycle continues to run in a cyclical pattern of around 24-25 hours, but that's then brought into line, i.e. it's entrained with exogenous Zeitgebers. So endogenous pacemakers help provide stability in the sleep-wake cycle, and Zeitgebers provide the means for adjusting this cycle when the environment changes in a significant way. For example, when you travel abroad and you're in a brand new time zone, your exogenous Zeitgebers help to entrain or retrain or bring into line your new sleep-wake cycle. And entrainment is desirable. If not, then we would just sleep through all of our holidays. And so we need this careful balance between endogenous pacemakers and exogenous Zeitgebers. So the two we're going to be looking at are light and social cues. So light is a key Zeitgeber in humans. It can reset the body's main endogenous pacemaker, the SCN, and therefore plays a really important role in the maintenance of the sleep-wake cycle. It also, interestingly, has an indirect influence on on other key processes like hormone secretion, blood circulation, etc. But we're really focusing on the sleep-wake cycle here. As we saw from the diagram previously, light information passes via the SCN to the pineal gland, where if it is dark at night, the pineal gland will increase melatonin production to induce sleepiness, drowsiness. And during the day, that light information will decrease melatonin production to increase wakefulness. Another key Zeitgeber is social cues. As every parent would know, babies rarely are on the same sleep-wake cycle as the rest of the family. In fact, newborn babies' initial sleep-wake cycle is pretty much random. At about 6 weeks, the circadian rhythm begins, and by 16 weeks, most babies' rhythms will have been entrained by the schedules imposed by parents, and this includes adult-determined meal times and bedtimes. Research on jet lag suggests that adapting to the local times for eating and sleeping, rather than responding to your own feelings of fatigue and hunger, is an effective way of entraining circadian rhythms and beating jet lag when travelling long distances. So there you go, there's a tip for you. Ignore your own cues for hunger and sleep and follow the social cues instead to help entrain your new circadian rhythm. All right, so let's evaluate these exogenous Zeitgebers. So we can use Seif's cave study here. We looked at this when we looked at circadian rhythms. And we could also use Seif's research here to show the importance of light as an exogenous Zeitgeber. You will remember from Seif's cave study that at first he had no real clear pattern in his sleep-wake cycle once he was in that cave, but he eventually settled on 25 hours. Each day he was waking up almost an hour later, and by the end he had lost a considerable number of days. Remember, he woke up in mid-September thinking it was mid-August. We can also use the bunker studies that we have looked at, where most participants settle on around 25 hours. So while this does provide evidence that there is an internal body clock, and it does show the importance of endogenous pacemakers, it also suggests that external cues, particularly light, that was missing in the caves, are important in training our circadian rhythms and bringing them in line with the 24-hour day. Otherwise, we would be free running at around 25 hours. Okay, so let's have a look at further research into exogenous Zeitgebers. So in this innovative study by Campbell, they demonstrated that light may be detected by skin receptor sites on the body, even when the information is not received by the eyes. So 15 participants, they were woken at various times by having a light pad that was shone on the back of their knees. The researchers managed to produce a deviation in the participants' usual sleep-wake cycle, up to 3 hours in some cases for some participants. And this shows just how powerful light is as an exogenous Zeitgeber, and it doesn't necessarily rely on the eyes to exert its influence on the brain. So again, just showing how powerful light is in that it can cause deviations of up to 3 hours in a sleep-wake cycle, even when shone on the back of the knees rather than the eyes. But, we must be cautious here. This research, despite now being quite old, has not been replicated. And some may argue that there were key issues in the actual study itself, where although they thought that the light was just shining on the back of participants' knees, actually there was surrounding light from the torch that may have made its way to the participants' eyes, which causes a confounding variable there. That makes us sort of question how effective light might be, particularly when the light information is not received by the eyes. Other key issues with research into exogenous Zeitgebers is that they don't have the same effect in all environments. The experience of people who live in places where there is very little darkness in summer and very little light in winter tell a different story from the usual narrative. For example, the Inuits in the Arctic Circle are said to have similar sleep-wake patterns all year round, despite spending almost 6 months in total darkness and 6 months in total light. So this suggests that perhaps the sleep-wake cycle is primarily controlled by endogenous pacemakers, and that these endogenous systems can override the environmental changes in light. So perhaps exogenous Zeitgebers have little bearing on our biological rhythm, or exogenous Zeitgebers have differing effects in different environments. Other research included a blind man who was born with a circadian rhythm of 24.9 hours, and he could not be entrained by mealtimes or other social cues. So social cues alone, therefore, are perhaps not effective in resetting or in training the biological rhythms. We need to do further research here on different individuals in different environments to see what effect exogenous Zeitgebers might have on moderating and influencing the sleep-wake cycle. Another thing to keep in mind as well is that all of this research into endogenous pacemakers and endogenous Zeitgebers involves isolating specific single Zeitgebers. So we isolate light or we isolate social cues. But that doesn't really give us much information on how endogenous pacemakers and exogenous Zeitgebers interact to influence the sleep-wake cycle. We're studying them in isolation. But as we know, it's an interactionist system. Pacemakers and Zeitgebers work together. They interact to influence our sleep-wake cycle. So it doesn't make sense to artificially separate the two for the purpose of research. So when we look next year at reductionism versus holism, then you will have a lot more to be able to sort of discuss and elaborate on that point. So just something to sort of keep in mind. All right, that's endogenous pacemakers and exogenous Zeitgebers, and therefore the end of biopsych.